It is my great pleasure to introduce our next speaker, Professor Carolin Schön. Professor Schön studied agricultural sciences at Hohenheim University in Germany and Oregon State University at the USA. After her doctorate, she served as director of the State Plant Breeding Institute at Hohenheim University, where she completed her postdoctoral studies in 2006. She has held a full professorship at Technical University of Munich since 2007. Her research activities are focused on the optimization of agricultural processes through the integration of molecular biology technologies. This primarily involves the use of molecular markers in breeding crops plants. Her work is also concerned with the characterization of native biodiversity and the identification of its functional characteristics, as well as the development of optimized breeding strategies for complex properties. Professor Schön is a member of the National Academy of Sciences Leopoldina, member of the Senate of the German Research Foundation, and member of numerous national and international scientific advisor councils. She played a leadership role in establishing SynBreed, an interdisciplinary network for genome-based breeding research on plants and animals. Professor, welcome.
Thank you very much. So thanks everybody for inviting me to this conference. Actually, as in the introduction, it was also mentioned, I do have a strong tie with animal breeding also, at least theoretically. So I'm still working with crops most, but the collaboration to animal breeders has brought me to collaborate with Dan Genola and a lot of other Uruguayan scientists. So that has enriched my scientific career tremendously, and I'd like to I'm very thankful for that. So hello to all the Uruguayans, or everybody really, but especially to all the Uruguayans who I have worked with and who I have met. And thanks, Ben, for a wonderful talk, and also that you kind of talked a lot about genomic prediction, because I will choose a slightly different topic, as I had predicted that Ben will talk about prediction. So my topic today is how to make maize landraces accessible for germplasm improvement. So I'd like to first acknowledge the collaborators in this project. It's a project that we have been working on for a few years now. I'm coordinating it, and so there's other collaborators, for example, the group of Henna Senjana in Göttingen, who is also an animal breeder. We have a private partner, KWS, and we're working on native diversity, trying to make it accessible for breeding and elite germplasm improvement. We have a website for it, so if you're interested in more. And I'd like to acknowledge the funding from the Federal Ministry of Education and Research. So the work I will be presenting is the work of mainly one of my PhD students, Manfred Meyer. So this is a picture of the Augenstein Research Station, where we have our field trials just outside Munich. And so this is like on one of our field days here. So in plants, the use of genomic resources is not really questioned anymore. We have very nice success stories, how genetic resources can actually help in improving elite germplasm. And of course, I mean, Jose Crossa and Simit have dealt with this for a long time, I suppose. One of the most prominent success stories is actually the Green Revolution, where the semi-dwarfing gene came from a genetic resource in wheat. So in rice, there is the submergence tolerance gene that is also very important in rice production. So the question, do we have to go back in time, can absolutely be answered with yes for qualitative traits. But can we also does it also make sense for quantitative traits? That has not really been shown yet. And for example, Bill Hill, a very famous quantitative geneticist, asked me, well, why do you even bother about genetic resources? Why don't you just make your populations bigger? I think that's a legitimate question now. So we took up the project to answer maybe some of these questions. So maize is an outcrossing species, and so it's quite similar to the populations are outcrossing, so quite similar to cattle, for example. They're heterogeneous and heterozygous. That means for us, they're very difficult to maintain and especially to evaluate, because we don't have data from production like the cattle breeders have. We need to put up trials for every genotype that we evaluate, and we need reproducible genetic units that we can reproduce and test them over replications and over many environments, because G/E is a big problem for us. And also we have quite difficult population structures, and we're not moving in a random mating population system. What also is, of course, problematic is that we have a huge performance gap between the genetic resources and populations that have been developed by breeding and improved by breeding for the last 50 years, at least. So, of course, the gap widens by the minute, but because of the breeding activities, whereas the resources just sit there in the gene banks and don't do anything except being maintained. So the production gap widens. And probably one of the worst things is that we have a big information gap. So we have started to characterize many of the genetic resources molecularly. So we have good molecular inventories, but it's very difficult to link these genotypes to meaningful phenotypes. And this is really one of the major drawbacks that we've had in using genetic resources so far. So there have been, of course, many attempts to start this. There have been breeding programs, but really the success for quantitative trait improvement and introduction of new diversity into germplasm, into elite germplasm, has not been as you would think with all what is possible with all the tools we've had. And so there is like now a lot of research going on in characterizing landraces. And just as an estimate, the elite germplasm in maize we're using represents only about 2% of the variation that is out there. So we think that 98% of the variation is not covered by the elite germplasm. That's an estimate that we have. So in a study by Ed Butler's group in Cornell, they looked at 4,500 landraces, and CIMMYT, of course, is contributing in a tremendous way to these activities. And so they looked at 4,500 landrace accessions, one heterozygous individual per landrace. And what they found is that they did a genome-wide association study on it. So they tried to map genes for adaptation and flowering time. And maize is especially dynamic because you can find it pretty much anywhere from north to south, from east to west. Has a very, very flexible dynamic genome. So there's a huge diversity out there. But there's also a lot of genes that contribute to adaptation, because a maize line that you grow in the highlands is not has a lot of different genes from those that you grow in the lowlands, or a line that you grow so south and north. You have photoperiod response, you have temperature responses, so it's very complicated to get really a good picture about all the genes that are contributing to flowering time, to adaptation. So this is a huge challenge, and it's clearly a quantitative trait when you look at the number of genes. And one of the big problems is that the mutations, the adaptive mutations, they co-map with the deleterious mutations. So it's difficult to get rid of the deleterious mutations and the mutations for other traits. So unless you can get rid of the adaptive mutations, it's difficult to discover the mutations that could be really novel and add to your germplasm. So then we took a different avenue, and what we said is we're focusing on very few landraces, and especially those that have been adapted over time through in situ population breeding to the target environments that we're interested in. And for us in Germany, this is Central Europe, basically. And so what we did was we said that, well, we will generate reproducible genetic units, and I'll comment on this a little bit, from few landraces, not very many, just a few. We will phenotype them for variation in their target traits beforehand, which is on an individual plant basis. And we try to minimize the background noise from the adaptive alleles and minimize hidden population structure, which is always a complication in genome-wide association studies. So then we took samples from 35 European maize landraces, and they're distributed all over Europe, from Galicia here in Spain to northern Germany and even further east. And we characterized about 1,000 of these plants with 500K SNPs. We're usually working with array data. 500K is a very large SNP that we array that we have in maize. And then we looked at the molecular diversity, and it clearly showed that about 95% of the variation is captured on average by 5 of these 35 landraces. So most of the variation is within the populations, not between the populations. So that kind of confirmed that we're going the right way. We had a clear population structure that we can define by pedigree. That helps in the GWAS model to define the population structure. And if we stay within a certain landrace, it's easier because the linkage phases, that means the association of alleles, positive alleles with a given marker allele, is a lot more consistent within populations than between populations. That's the red line here. And the linkage phases, that you have the same linkage phases, the correlation between linkage phases is much higher within populations. And the LD, so you need, of course, very good resolution in the genome-wide association mapping. And the LD you can get when you're taking, let's say, 400 individuals from one landrace is already very high. And this here shows what you get if you kind of combine one individual from 35 landraces, or you combine 10 individuals from less landraces, or if you just use one landrace and take the sample out of there. And what we find is that the resolution becomes already very good with few landraces also already, and a fairly large sample within a landrace. So that encouraged us to move on. And the target trait that we were interested in maize was early development. Coming back to the climate change issue, we want to kind of plant earlier because we need to evade the drought season and the heat during flowering. So we want to move on with the planting earlier, but then we have the problem of cold and other like limitations. So basically what we want is full coverage here of the ground as soon as possible, less nitrogen leakage, less erosion, then less herbicide treatment. So it has a lot of sustainability effects if you get very, very fast early development. This is a complex trait. It's a combination of like cold tolerance, vigor, germination. So it's probably determined by many genes. And I will talk about something called the Flint elite material. Flint is a heterotic group that we use in Central Europe. And so we're staying with one heterotic group. We're talking about hybrid breeding, hybrid crops, and the population improvement happens within one heterotic group. So I will stay within this one heterotic group. And all the landraces fit to this heterotic group. And of course, as we know from quantitative traits, that comes along with all these complications, many genes, small effects, background, and environment dependencies. And especially for this trait, as you can see, this is Manfred, the PhD student, and Victoria, one of my postdocs. It is not fun to assess the trait. So then we did a phenotypic pre-selection, and we looked at variation within landraces for the target trait, and then we chose 3 landraces for which we had a fairly large within landrace variance. And here you already see like the variation that we're looking at, even though we're staying within the same heterotic pool. So you can imagine what it's like when you go across pools, tropical, subtropical, moderate, temperate material. It's huge. So then we selected 3 of the landraces, and 1 from Galicia, 1 from Austria, and 1 from northern Germany. They represented different groups here in this neighbor joining tree, and then we moved on with that. And then the first step was to produce reproducible genetic units. Well, in maize, we are lucky because we have a system that we can produce very quickly fully homozygous lines. So in maize breeding, we work a lot with inbred lines for the development of hybrids. And then it's important to become homozygous very, very fast. So what we're doing is we take these landraces, these 3 landraces, we're pollinating them with a special maize, we call it inducer, and then we collect haploid seed on these plants. And then this gives us haploid plants. We double them with colchicine, and then we have a doubled haploid or a plant that is fully homozygous. Then we multiply the seed, we genotype it, and then we have 3 landrace DH libraries with about 1,000 lines. And as Ben already pointed out, of course, you want them to be as big as possible. But I mean, collecting haploid, double haploid plants from landraces is hugely expensive, because the efficiency of double haploid production is about a factor 10 less than what we have in elite material. So it's very, very cumbersome to do this. And not all landraces are amenable to double haploid production, I have to say that. But when you look at this, it has many, many, many advantages. What you see here is a sample of diversity that we have in these libraries. And I think for those who are animal breeders maybe, or not so familiar with maize breeding, so 1 row is 1 double haploid line. And you can see one plant is like the other. So phenotyping is very, very easy in these double haploid lines. And what you also see is that the huge variation that we have. So in this population, we have twice the additive genetic variance because we're fully homozygous. So, and it's quite a lot of variation you get from landrace. And I think it also shows that with those 3 landraces, we do have a very large variation. And then we go about and test these lines in field trials. Again, here, this is again what I've just shown you. So we usually lose some on the way. So we tested them in up to 11 environments, 2 years, as lines per se, so the inbred lines. But then in hybrid breeding, of course, we're interested in the hybrid value, the breeding value for yield, basically, of those lines. And then that means that we always cross it to a line from the opposite heterotic pool. And in this picture, you see a little bit what happens when you do this, because up here in the corner, on the top left corner, you see the test crosses. That is, it's exactly the same lines, but they're crossed with a tester. You can already see that you only have less variance here, and a lot of the tester allele covers a lot of the variation, but that's the data we need to collect on yield if we want to make prediction for hybrid, for general combining ability or breeding value for yield. So we have test crosses in up to 7 environments, data on that, and I will now show you some of the results. Before I do that, I give you some impressions of what those landraces look like. So this is the outcome. This is 1 double haploid line, and you see that these maize lines don't even show their tassels. My students call this the horror picture show of landraces. Of course, that's something you cannot use. But of course, you can map the genes that are responsible for these traits. So this is a rust epidemic. We had tested them in Spain, and all of a sudden we got a problem with rust. Of course, this is also a phenotype you wouldn't want, but it's like a mutant collection. So this will probably keep me busy till the rest of my scientific career. You have lots and lots of traits you can map in these populations and clone. This, for example, is a trait that you have like very small plants, very large plants, and so that's of course something you certainly don't want. The trait we were after, it's seen, you can see it right here. So you have some lines that are much higher than others, and it's of course those that develop very fast that we were interested in. And at the moment, we're looking at this with UAVs and imaging methods, so to make the phenotyping much faster. So this will be the trait I will be talking about from now on, differentiation in early development. And again, I think you can see the beauty of double haploid lines for differentiation. So were we successful in improving our target trait? That's test cross data, because I'll also show you some yield results in the next slide. And so what we see here is the results from 2 of those 3 landraces that we can see. I'm not showing the third because the numbers of the H lines was rather small we got on that, so I'm working only with the 2 larger populations. So what we see here is plant height at an early stage in centimeters, and basically what this shows is this is the distribution of the trait values for the 2 populations, the 2 landraces, the H lines from cross to a tester from these 2 populations. And what we see is that they definitely outperform what we call the founder lines, which are here, those black triangles, except maybe this one that is known for its very good early development. And they outperform commercial hybrids. So we can check the kind of mission accomplished. So we do have transgressive segregation over what we have. Because really that is what's important, right? You don't want the same over and over again. You want something new and something better. That's the idea. The picture is slightly different when you look at the test cross data and the yield performance. This is again the same 2 populations. These are the founder lines. These are the commercial checks. That kind of tells you what breeding progress means. We have about 20% yield gap between the mean of the DH lines and the commercial test crosses of the DH lines and the commercial checks. If we take the best 10% of these lines, we still have a yield gap of 10%. And I will not comment on this today because due to time, but of course, we're also looking at whole genome approaches, and of course, we're also looking into genomic prediction. And actually, I can already say that the predictive accuracies are very, very high, and we will be able to go through very rapid cycling with whole genome prediction to improve this. The question is for us, when does the calibration break down and where do we get the new calibration from? But whole genome prediction will hopefully solve this problem here. So then we were interested in discovering haplotypes that are favorable for the trait that were our target trait early development. So what we did is we constructed haplotypes from SNPs, and we chose a method, and I'll comment briefly on that in the next slide, just taking windows of 10 SNPs. So we constructed haplotypes and we coded them in their presence and absence. So if a haplotype was present, it got like a 0 or a 1, and if it wasn't present, it got a 0. So and then we ended up, then we filtered for linkage disequilibrium and minimum frequency, and then we ended up with about 150,000 haplotypes in our population. So actually the haplotype construction is, I think, the ultimate challenge in this procedure, because what is a haplotype? We don't really have a good answer for this. And so there's different ways of constructing haplotypes, and it's definitely not trivial. So, and I have to say the best method really depends on your study objective. There's different approaches. Some are like predefined markers like we did, predefined by the physical length. You can say you construct them by 100 kb windows or 1.1 centimorgans, whatever, you can like predefine them. But that kind of course, is arbitrary, right? How big do you want to make them? Then you have population-specific ways of doing it. You can do it through minimum R-squared distances. Excuse me. You can have limited recombination. You can have limited diversity. There's also flexible approaches like, just a second. There's flexible approaches just like what was constructed by the group in Göttingen. They do an IBD-wise haplotype construction, but because you don't have a fixed starting and fixed endpoint, this is not really useful for the application we're looking for. Because what we're looking for is, we're looking for haplotype trait associations, and we want to ask the questions, are those haplotypes present in the elite germplasm? So the problem we have is, if we make them too long, of course, they're broken up by recombination. If we make them too short, the extremist just snips. So I would only have two types, and of course, I would find one or the other one in the elite germplasm. And this method on the right doesn't really also give me a very good approach to really compare between the genetic resource and the elite germplasm. So these are the middle ones, the high LD and limited recombination are also population specific. So really, if you want to move across populations, what the best we have at the moment are these fixed windows. And we're currently working on, and that's really interdisciplinary research that really needs to be done, and we don't really have very good solutions for it yet. How can we use gene annotation, gene boundaries, the pan-genome idea for defining these haplotypes? Very important field of research in maize, at least. So after that, we performed a straightforward genome-wide association mapping study. We had a univariate model. So the target trait was entered into the bivector. We had a definition for like a fixed effect for the landraces. Then we had the fixed effects for the haplotypes. And then we had a random component for the genetic background. Then we came up with candidate haplotypes, and then we entered those into a multi-locus, multi-environment model, where all the information from the 11 environments and all the SNPs we had identified, all the haplotypes we had identified earlier, go in, and then came up with the final set of haplotypes.
Chris, I know you started late, but we are getting towards the end of the time space for your talk. So a few minutes more, finish.
All right. So, let me go through this then very briefly. So we came up with these trait-associated haplotypes. We found many of them for the traits we were interested in, very few for flowering time. So that was a good result. And then we looked at how many haplotypes do we find across environments. We have like the blue and the red for favorable and unfavorable. And of course, this is a typical picture that we see for haplotype effects across environments. So then we asked the question, are these haplotypes also present in elite germplasm? And so what we did was we looked at a random set of haplotypes, and you see here the gray curve. And this is a distribution of haplotype frequency in the breeding lines. We looked at 75 breeding lines for haplotypes that were randomly chosen. For the favorable ones for early development, we found some increase. For the unfavorable ones, we found very similar distribution as for the random ones. So what this shows us is that, okay, breeding did its job, but we also have some, and that's here at the 0 line, that are not present, that are favorable, but not present in the breeding lines. So the effects in breeding lines, you can see here. What we see here is the early development trait again, and we have like one haplotype. And here you see like the breeding line, the distribution of the trait value in the breeding lines. And what here in the dark green, you see all the lines from one of the landraces, and the light green is those that have what we say our focus or our favorable haplotype. And as we can see, the mean of the breeding lines overall and of the focus haplotype is significantly different. And here is another example where we see the same. So we believe these are novel haplotypes for a quantitative trait that we can actually use for germplasm improvement. Of course, we need to validate this by crosses. And we have a proof of concept. We take this with us. So in maize, we have a tillering gene. And with this, we also kind of validated our approach, and we see exactly the same there. And that kind of really tells us that the methods we chose does in fact help us to identify quantitative haplotypes that are novel for quantitative traits. So to summarize, we have novel haplotypes that are associated with target traits. We have a stability of allelic effects across environments and in populations. And important is also that the haplotypes that we find, they're not showing much pleiotropy or linkage with undesirable traits, because of course we have all these difficult traits in the landraces. And one thing that I haven't been able to show, but is that novel favorable haplotypes usually come at low frequency. And that's the major problem, of course, in the power of GWAS. And as I already mentioned, the haplotype definition is not really resolved. And with this, I'd like to finish. Landraces can serve as a novel variation. We can do a targeted introgression of favorable alleles. We can get rid of unfavorable alleles, and we can use whole genome breeding strategies and also maybe the cloning strategies to improve our elite germplasm with through pre-reading and introgression of new alleles. And with this, I'd like to thank again the funding agencies and all my collaborators for a good collaboration. Thanks a lot.
Thank you very much, Dr. Schön. We have time for one question. Daniel Janola, you can open your microphone.
Thank you very much. Guten Tag, Chris. Thank you very much for the very interesting presentation. I think you failed to mention that in your interaction with Uruguayan breeders, you had to update your football knowledge.
Oh, that's right. I always read on football before I meet with Janola, right? That's right.
No, but for a serious question, you mentioned that this question of haplotype reconstruction is a bit very much up in the air. And I'm curious, has there been any attention of construction haplotypes using multi-locus linkage disequilibrium? 3, 4, I think it's a highly neglected area.
Well, I think we both know, Dan, that multi-locus LD is, well, I talked to Bill Hill about this once, and he said it's like, well, combine it with epistasis, and it's a black hole going into a black hole, right? So it's a very difficult subject, yes, but yes, of course, it should certainly be helpful. I think we should combine bioinformatic information on the genomes, and of course, like LD and other theoretical points to get closer to solving the problem. Yes, I do believe so.
Thank you.